let me start recording and share my screen. All right, today is January 18th, and we're supposed to be covering chapter 2 and finishing it and starting chapter 3. It is too late to change the quiz on Wednesday. So for this week, the quiz will be on Wednesday. But in the future, if you guys want to change the date that quiz is held, we can discuss that, and I will make a discussion page in this week of the lecture to discuss this topic. Okay. All right. Any questions about what we're doing? I think I can close that down. Close that down. Let me call up lecture 2. Any questions about any of the lessons that we've covered?
Are you going to make chapter 2 PowerPoint available after tonight?
If you remind me in the lab, I'll do it in the lab.
Okay, thank you.
All right, so the last topic we talked about were lipids, and they consist of carbon, hydrogen, and oxygen. And there's very little oxygen in lipids. There's simple lipids, complex lipids, and steroids. Let's talk about simple lipids. They're made up of fatty acid and glycerol. We'll show a picture of that in just a minute. Fatty acids are major components of lipids. They consist of chains of carbons. Have we talked about this topic? No
We
comments?
started to.
That's where we started.
Yeah.
Okay, so we did talk about this. Did we talk about this slide?
No.
All right. So fatty acids can be saturated, and that means it's a single carbon link. All the hydrogens that are possible are attached to the carbon, or it can be an unsaturated fatty acid, and that has a double carbon bond, meaning 2 covalent bonds between the carbon. And wherever that happens, there's a kink in the chain. Let me blow this up. A double carbon bond here, and it creates a chink in the chain. And when we say all the links with carbon are possible on a saturated fatty acid, like here and here, right here, as you see, there's only one hydrogen. And to get another hydrogen there, you have to break that double carbon bond and then attach a hydrogen here and a hydrogen here. This is glycerol shown here. And then there's the fatty acid, and water comes out in dehydration synthesis to make that link, where there is a little bit of oxygen in this lipid, but there's only 2 oxygen atoms in a triglyceride, a molecule containing 1 glycerol, covalently bound to 3 fatty acids, why it's called a triglyceride. And this is the most common fat we have in the body. It's also the body's richest energy source, and it's stored as adipose in animals. And you can see triglycerides if you look down at your belly. Any question about any of that? All right, so that's a simple lipid, a triglyceride. We also have another type of lipid, and it's a phospholipid. This lipid is unique among lipids because lipids generally are hydrophobic, meaning they don't dissolve in water. They're non-polar. The phospholipid is polar on one end, which we call the head, and non-polar on the other end, which we call the tail. The tail is composed of two fatty acids. And yes, in humans at least, one fatty acid is always saturated, the other is always unsaturated. And on the head, there's the polar group, which contains glycerol, a phosphate group, which makes it polar, and then an organic group, which is the rest of the molecule. And we just abbreviate that as R. And we'll talk about R being the rest of the molecule in other molecules we talk about, like proteins. Any question about that? So the head is polar because of the phosphate group, and it actually is charged. And then the tail is nonpolar. And that's what the molecule looks like. And when we put these molecules together, especially when they're in water, they'll start lining up like this. That's one for humans. That's an unsaturated fatty acid, which you might find in, let me see, something that lives in the Arctic or in certain bacteria, especially archaeobacteria. Although that's not in the cell membrane, but and then in animals, for our cell membrane, we will see the membrane shaped like this, and inside the fatty, the phospholipid molecules, there will also be cholesterol. And the sterol molecule, which is cholesterol, gives strength and support to the membrane, the cell membrane. Any question about any of that?
Yeah, so what does it mean for it to be biphasic?
Biphasic, I haven't talked about that yet.
Okay.
Biphasic means that one end of the molecule is polar, the other end of the molecule is nonpolar, and this region of the molecule can dissolve in lipids. So this region of the molecule is hydrophobic. This region of the molecule can dissolve in water. So this region of the molecule is hydrophilic. And that makes this molecule biphasic. It can dissolve in both oil and in water. There aren't too many molecules which have properties like that. There is one that you know of, detergents. And it's because detergents are biphasic, meaning they dissolve in water and they dissolve in oil, that detergents help wash our clothes better than water alone, meaning water can dissolve the water-soluble molecules and make the clothes cleaner in that regard, because just the water is cleaning it. And then the detergent, when you add it, can dissolve in the water because of this region here. And this region can dissolve in the oil that gets in our clothes. And then the detergent can clean our clothes. Any question about any of that? I was trying to look, I can't see it for some reason, because maybe this is in the way. It's unusual lipid because it's biphasic. All right. When we put phospholipids together in water, they start forming this way because this part of the molecule likes to face the water this way, and this part of the molecule likes to face the water this way. And then the hydrophobic tails come together because they don't want to be around water. They don't like being around water. And that will make the phospholipids form this shape, a liposome, where the heads are facing the water outside, and then the heads of the phospholipid are facing the water inside. And then the hydrophobic tails of the phospholipids come together, and you'll see this is a bilayer. There's one phospholipid layer here, and then another phospholipid layer here. And this should look familiar to you. What does that look like? Come on, what's round, has water on the outside, specifically if it's in the blood, and has water on the inside that we call cytoplasm. Nobody's gonna guess? All right, we'll move on. You can also have phospholipids inside of water forming this, and that's much less interesting to us than that. This is actually what the cell membrane looks like, a phospholipid bilayer with heads facing the inside of the cell because it's mostly water in the cytoplasm, and then phospholipid head facing outside the cell because the cell is mostly, on the outside of the cell is mostly water, and you're mostly made up of water. And think of it as a blood cell, the cell membrane on the outside of the cell, it's going to be mostly water. And then this bilayer forms the layer controlling what comes into and out of the cell, meaning it's the phospholipid fatty acid tails of the phospholipids, which make the active ingredient of the cell membrane, which allows things to come into and out of the cell. Any questions about any of that? All right, I'm not hearing anything, so I'm moving forward. Let me try putting that. That might work. Up on the top. Steroids are four interconnected rigid carbon rings shown here that characterize this group, and they form the steroids. If there's an OH group attached to the steroid right here, we then call it a sterol. And cholesterol is the most important sterol in our bodies. Sterols are an important part of the animal plasma membrane. And the sterol, meaning cholesterol, gets in the plasma membrane to help give it strength and support, because that's all animals have to protect their cell, and that's the plasma membrane. Plant cells, of course, have a cell wall outside of the plasma membrane, and that cell wall protects the plasma membrane. Any question about any of that? All right, can somebody confirm that I have you guys here? I'm not seeing anybody, although the chat says that somebody's here. Okay. Cholesterol is only found in what foods that you eat? And what food is it not found in? Nobody knows when you eat something whether it has cholesterol in it or not? I'm shocked that nobody knows that.
Is it
Cholesterol,
grains?
well, a brain has a high proportion of cholesterol, but what I was trying to get at was animal tissue or animal products have cholesterol. And if you eat plant matter, like, I don't know, broccoli, cauliflower, carrots, there is no cholesterol in plant products, including bread. So you only get cholesterol when you're eating animal products like bacon or sausage or hot dogs or anything that's made of meat. And eggs and milk. Any question about any of that? Let me remind you that lipids, although some of them are made up of monomers, like the phospholipid has a phosphate group, and then the fatty acids, the triglycerol has a glycerol molecule, and then a 3 fatty acid groups. So these would be the monomers. And then this would be not really a polymer because 3 and 4 isn't really a polymer, but it's a larger molecule. But the steroids and the sterols are not made up of smaller molecules. So they're not composed of monomers. Do you guys get that? So the lipids, some of them are composed of monomers, but some of them are not. And all of the other biological molecule, all of the major, meaning the big biological molecule that we're going to call a polymer, is made up of monomers. All right, let's move on to proteins. Proteins are essential in cell structure and function. Proteins have a lot of diverse function and what do you call it? Structural roles in cells. Proteins may be structural proteins found in bone, muscle, or hair. Proteins may be enzymes which speed up chemical reactions. We'll talk about that in a little bit, not today, but later in the class. Proteins may be transport proteins, which move chemicals across the plasma membrane. Proteins may be regulatory proteins, which regulate body functions. An example would be a hormone, like a growth hormone that causes the child to grow. Proteins may be responsible for movement, like if a bacteria can swim in a liquid media, like a broth, it's happening because of a protein. And then we move because of the proteins in our muscles. Proteins are also responsible for some bacterial toxins. And then lastly, proteins can be antibodies. And these are proteins that the body makes to help defend itself. Any question about any of that? All right, proteins, all of them consist of monomers, and these monomers we call amino acids. Amino acids have an amino group on one end of the central carbon and a carboxyl group on the other end. So that means proteins are always polar because the amino group is polar and the carboxyl group is polar. There's always a hydrogen attached to the carbon on one end of the carbon, and then an R group on the last link that binds to carbon. The R group is just the rest of the molecule, and there are 20 different R groups that humans have. because there are 20 different amino acids. On which amino acid it is depends on which R group it has. So R is just the rest of the molecule. You should know this general structure for an amino acid. And here is one R group for the amino acid tyrosine. And you'll note that this region of the molecule is nonpolar, and then this region is polar. So proteins are always polar or hydrophilic from the amino group of the amino acids in the carboxyl group, and some of the R groups have polar regions to them. So proteins are always polar, and proteins are always nonpolar because some of the R groups have regions which are nonpolar. Any question about any of that?
So they're both polar and nonpolar?
Yes, proteins are always both polar and nonpolar. We'll talk a little bit more about that in a minute. Proteins are made up of amino acids, and you have many amino acids which are linked together to form the protein. As I mentioned, there are 20 different amino acids. You don't need to memorize this slide or the next one. These are just showing you the different R groups. The simplest R group is simply hydrogen, which is in the amino acid glycine. And so that R is simply a hydrogen. This R group, I'm just showing you this one because it's a nonpolar R group. It has a methyl group attached for the R group. And this one is 3 methyl groups attached together, and it's nonpolar. Well, all of these are nonpolar R groups. This is a nonpolar R group. Like I said, this is a partially polar R group. This part of the molecule is polar. This part of the molecule is not, this part of the R group is nonpolar. This part is polar with the amino group here and this amino group there. This carbon here, that carbon there, that carbon there, and that carbon there are nonpolar. Anyways, you're just seeing some of the different R groups. Amino acids are linked together by peptide bonds. That is where the OH comes off of this amino acid and the hydrogen comes off of this amino acid. And then this nitrogen directly bonds to that carbon of this amino acid. And that happens in a dehydration synthesis. Water comes out, and then that nitrogen bonds directly to this carbon here. And this we call a peptide bond because this bond is seen in peptides, meaning proteins. Peptide is a molecule like a protein, but it's smaller than a protein. We'll talk a little bit more about that in a minute, where the nitrogen bonds directly to the carbon. It's not found, this bond, very often, both in our body, the peptide bond, or in the universe, but it is found in proteins, which is why it's called a peptide bond. And then when many amino acids link together by peptide bonds, we get a polypeptide, which is made up of monomers. And a polypeptide just means many peptides or many amino acids. And when you add the last amino acid to a growing protein, that's where we no longer call it a polypeptide, and we call it a protein. Everyone get that? So when you're first building a protein, you get one amino acid and you join it to another amino acid. This we don't call a protein. And it's not really a polypeptide because it's only two, but it is a peptide. And then you just keep adding on more and more amino acids. You get a polypeptide. And then when you add on the last amino acid, that is when we call it a protein. Any questions there? When we're talking about proteins, proteins have 4 levels to the protein structure. There's the primary level, the secondary level, and the tertiary level, and then lastly, the quaternary level of a protein structure. This is loading slowly. The primary level is just the protein.
This is your living room / yoga shanty / regional office / and this is the basement / panic room. Maybe what your
We
family
have a
needs
commercial
is a vacation
here.
home / vacation.
Here we go.
In this video, we're going to talk about the structure of proteins. And what basically is a protein?
For some reason, we're not seeing it.
A protein is a polymer consistent of many amino acids. So each amino acid represented by the circle is a monomer that forms the protein. Whenever you have many amino acids, it's also called a polypeptide. And the bond that connects each individual amino acid residue is a peptide bond. So let's talk about the individual structure of an amino acid. So an amino acid has a chiral carbon with a hydrogen attached to it, and it has an amine group attached to it as well. And then it has an R group and a carboxyl group. Now I'm going to draw another amino acid right next to it. The left side, where the nitrogen is located, is the N-terminal of the amino acid. The right side, that has the carboxyl group, is the C-terminal of the amino acid. Now, these two amino acids can react with each other in a condensation reaction where we're going to lose water, and we're going to connect the two amino acids into one molecule. So once water is removed, we could form a peptide bond. This is also called a dehydration reaction because you're losing water. And so this is going to be the new product of the reaction. So let's see if I can fit it in here. So this oxygen is gone. So now we have a nitrogen. Let me take out H2O. We have one hydrogen left over. We lost two once we lost water. So this is a dipeptide because we have two amino acids combined. And so this amide bond that we see here, that is the peptide bond. We formed a bond between the carbon and the nitrogen atom. So whenever you combine two amino acids to form a peptide bond, you're forming a covalent bond, which is hard to break. Now let's talk about the different levels of protein structure. There's 4 levels that you need to know: the primary structure, the secondary structure, the tertiary structure, and finally the quaternary structure. Now the primary structure is simply based on the sequence of the amino acids found in the protein. The sequence determines the shape of the protein. If you replace just one amino acid with another, it will completely change the shape of the protein. So the shape and its function is primarily determined by the sequence of amino acids. The secondary structure describes the localized shape of a protein. And there's two of them that you need to be familiar with: the alpha helix and the beta pleated sheet. In the alpha helix, you could see that it's stabilized by hydrogen bonds. The NH group of one amino acid interacts with the carbonyl group of another amino acid. And each turn contains about 3.6 amino acid residues. Now here is a visual illustration of the beta pleated sheet. Just like the alpha helix, it too is stabilized by hydrogen bonds between the carbonyl group of one amino acid and the NH group of another amino acid. Next up, we have the tertiary structure, which represents the three-dimensional complete folding pattern of the protein. And so here's a visual illustration of it. So we could see some areas is just a straight chain. Here we have an alpha helix, and here we have a beta pleated sheet. Now, the tertiary structure is one individual subunit. When you combine multiple subunits, you create a quaternary structure. So hemoglobin is an example of that. It has 4 individual subunits, 2 alpha subunits and 2 beta subunits. So hemoglobin is a protein with a quaternary structure. And so that's basically it for this video. Hopefully it gave you a basic understanding of proteins and the 4 levels of structure that they have. Thanks for watching.
Pop-Tarts will start Sunday Fun Day on.
All right, any questions about that? So the primary level of protein structure is simply the sequence of amino acids. And that's all the protein has when it's first made. But the amino acids start interacting, and that gives rise to the secondary structure. Let's see, we already talked about that. What happens is that the nearby amino acids, the amino acids start interacting with other nearby amino acids. So in the secondary structure, you have interactions between neighboring amino acids. So the secondary structure, secondary structure is the interaction between neighboring amino acids. And there's a lot of interactions that can happen. I think the video may have only said that there was one. And your textbook may say there's only one. And that is, there's a hydrogen bond that can form between this amino acid and that amino acid. But there are other interactions that can happen in the secondary structure. And that is, there can be repulsion between this amino acid and that amino acid. This could be a hydrophilic amino acid, this is a hydrophobic amino acid, and they will repulse each other. These are both polar and they form a hydrogen bond. But then you can have interactions between 2 amino acids. Let's do this one because there's no hydrogen bond shown. You can have this one nonpolar and that one nonpolar. These two will like to come together by van der Waals forces. We're not going to talk much about that, but nonpolar amino acids like being around other nonpolar amino acids. And so they will be attracted to each other. You can also have an amino acid which is positively charged, like an arginine, and then an amino acid which is negatively charged, and they will be attracted to each other by ionic attraction, meaning the positive charge is attracted to the negative charge. There can be repulsion. If this one is positive charge and that one is positive charge, they would be repulsed from each other. Let me see, have I mentioned anything else? I think that's about it for the different types of attractions and the different types of repulsions. I might not have mentioned that if this one is nonpolar and this one is polar, they will be repulsed from each other. Any questions about all of the different types of interactions that can happen in the secondary structure? Now, I won't ask you, but there can be different types of secondary structure. There can be an alpha helix, as shown here, and shown here, where the protein starts, I don't know, coiling like a spiral staircase. There can be a random coil, which isn't shown here, but it's sort of shown here, a random coil. And then there can be a pleated sheet, which is shown here. Let me see if I can get a little more of that on. Where we have the protein, I don't know if it's running this way or running that way. And then there's multiple attractions between the amino acids, and that they call a pleated sheet. Any questions about the secondary structure? It happens from the nearby amino acids being either attracted to each other or repulsed from each other. But the point is, it's always nearby amino acids. And the tertiary structure, we have amino acids interacting with other amino acids, but they can happen all over the protein. Like in this interaction here, we have an amino acid interacting. Well, actually, let's do it for this one here. We have an amino acid near the end of the protein chain interacting with an amino acid at the beginning of the amino acid chain or the protein chain. So tertiary structure is the same as secondary structure, except that the interactions between amino acids can happen all over the protein chain. It's not just the nearby amino acids interacting. Any question about that? And what are the amino acid interactions? They can be the same you see in the secondary structure. They can be hydrogen bonds forming between two polar amino acids. They can be non-polar and a polar repulsion, or they can be ionic attraction. This is an ion positive amino acid. This is a negative amino acid. They'll be attracted to each other. They can be polar and non-polar, or they could be both polar here, and they'll be repulsed from each other. There's the van der Waals forces, where two non-polar amino acids will be attracted to each other. And these attractions can be all over from amino acids all over the protein chain. And then there can be non-polar repulsions. Let me give an example, like maybe this amino acid is repulsed from this amino acid, and that could be from ionic charges. This could be positive, that could be positive. It could be from non-polar and polar. They will be repulsed from each other. Is there another repulsion? Not that I remember. And I think I talked about the attractions. There is one attractive force which is seen in the tertiary structure, and it is not seen in the secondary structure. In the tertiary structure, you can have sulfhydryl bridges or disulfide bridges forming. What happens is, where do I have that shown? Oh, here it is right here. Let me blow this up. Between the amino acid cysteine, cysteine has a sulfur. Let me show this in a new window. Cysteine, most of the amino acid is here, and it has a sulfhydryl link. I should probably make that capital. And that can get together with another cysteine, which has a sulfur hydrogen link. Let me get that on the other side here. Ah. And these sulfhydrogens can get together where the hydrogen comes off, going into solution, which forms an acid, by the way. And then we have a covalent link between these sulfurs. And that's a covalent covalent bond between these sulfur groups. And that we call the disulfide bridges. And it's a very strong linkage. The hydrogen bond, as you know, is a very weak linkage. And an example of the disulfide bridge and how much stronger it is, when you get your hair permed, it makes the disulfide bridges in your hair. And you can go into the shower and wash your hair for a month, and you'll still have your hair permed. Although after a month, the perm will start to fail. The hydrogen linking in your hair forms when my mother got her hair, she would put it in curlers and then put on the hair curling cap and then turn on the hair curling. I think it put steam in her hair. And then she would stop it after, I don't know, some half hour or 10 minutes, and then take out the cap and take out the curls. And she would get curls in her hair, which would simply be a hydrogen bond form. And the trouble with that is she would then go out into the Oregon mist or the Oregon rain, and the rainwater would get in the hair, and that would immediately break the hydrogen bond. The point is hydrogen bond breaks and reforms all the time, and it's a weak link. And this covalent bond in the disulfide bridge is a very strong bond.
Is the
Any
disulfide
question
bridge
about
the
any of that?
reason why Black people have like really curly hair?
Sorry, can you say that again? I didn't hear it.
Is the disulfide bridge the reason Black people have like really curly hair?
Not entirely. Like I said, when people get their hair curled, what do you call that? Permed, it does put in the curls. And it is true that Blacks do have some disulfide bridges, but Blacks can get their hair permed as well, meaning that they can get more of them. So yes, blacks have those disulfide bridges in their hair. And I don't know if whites do or not. So,
I have a
so did I
question.
answer your question? And I guess I can say partly. Okay, but blacks can get their hair permed as well. I don't know what it looks like when they get their hair permed, but they can get their hair permed.
So what is the only difference between the two is that there is the irregular helix folds?
Between white hair and black hair?
No, secondary and
Oh, yeah, there's two differences between tertiary structure and secondary structure. One, in secondary structure, the interactions between the amino acids happens only between neighboring amino acids. In tertiary structure, it can happen between any two amino acids. In tertiary structure, another difference is we have disulfide bonds forming. You don't see that in secondary structure. Okay.
okay, thank you.
For some proteins, that was an excellent question, by the way. Sorry, I got to straighten this. Between some proteins, all they go up to is tertiary structure. Any protein that is made up of one polypeptide unit only goes up to tertiary structure. And for my knowledge, that's all enzymes, but I'm just going to say that's most enzymes. They only go up to tertiary structure. However, if the protein is made up of more than one polypeptide unit, these polypeptide units have to come together and form the quaternary structure of the protein. So if the protein is made up of more than two or more polypeptides, they come together. And then they will bond together, meaning you get all of the links occurring in the quaternary structure that you see in the tertiary structure. And that is mainly hydrogen bonds, van der Waals forces, ionic bonds, ionic repulsion, polar and nonpolar repulsion, and then the disulfide bridges. Some examples of proteins that have quaternary structure: hemoglobin, insulin, and antibodies. And all of these proteins are made up of, all the ones that I mentioned, are made up of 4 polypeptides that come together to form the protein, the final protein. All right, any question about that? So some proteins have a quaternary structure or a quaternary level of the protein structure, but not all proteins do. If the protein is made up of only one polypeptide unit, then the highest it can go is a tertiary structure. All right. Proteins have different shapes. If the shape is generally compact and roughly spherical, we call it a globular protein. Hemoglobin is a globular protein, as is most enzymes. There can also be fibrous proteins, and they are thread-like. And molecules or proteins like collagen are fibrous, thread-like. Proteins are a little different than other of the biological molecules, and you can get conjugated proteins. And these are combination of amino acids or proteins with other organic or inorganic components. We name these non-protein components by the non-protein part, meaning a glycoprotein is a protein and carbohydrates called glycoprotein, and a lipoprotein is a protein bound together with lipids. Any question about any of that? You see glycoproteins in your blood and on the cell membrane, and that's true with lipoproteins, which most of you might be familiar with lipoproteins in your blood. Proteins can become denatured, and that is when the protein loses its three-dimensional shape. When the protein starts breaking the hydrogen bonds between the protein or forming hydrogen bonds that are not normally there, the protein can take on a different three-dimensional shape and it can start to lose its function. And when it starts to lose its function, we say it is becoming denatured. And when it no longer has its function, we say it is denatured. Proteins can become denatured when the protein is at its incorrect temperature, at its incorrect pH, or incorrect salt concentration. Let me talk just a little bit about that. An example of protein denaturation is when you take an egg whites are a protein, and when they're runny, the protein is not denatured. And then when you cook it, you denature the protein. Another example of a protein denatured, which you probably did in your preliminary biology class, is you take an enzyme and you move it to a temperature that is not optimal. When you move away from the optimal temperature, the enzyme starts losing its function, so it won't work as well. And then when you move it to a temperature that is way away from the optimal, such as freeze the enzyme, you denature it, and that actually, when you freeze it, you start forming hydrogen bonds in the protein that shouldn't be there. And then when you heat the protein, you also denature it, and that breaks the hydrogen bonds that should be there. And you can heat the protein and denature it. Any question about any of that? So proteins are more complex and diverse than the three other biological molecules. And that's because they have many different building groups. Instead of just a few or a handful of monomers, humans have 20 different amino acids for the monomers which make up proteins. Any question about any of that? All right, let's talk about our last of the biological molecule, and that is nucleic acids. Nucleic acids, there are two: DNA, or deoxyribonucleic acid, and the second being RNA, or ribonucleic acid. Nucleic acid consists of monomers, which are the subunits that we call nucleotides. A nucleotide itself has three parts to the molecule. So nucleic acids are made up of nucleotides, and nucleotides are made up of three parts. There's a pentose sugar, shown here, pentose meaning 5-carbon sugar, and that is always either ribose in RNA or deoxyribose in DNA. And it's the sugar that gives deoxyribonucleic acid its name, or ribonucleic acid. The second part, besides the sugar, is nucleotides have a phosphate group shown here. And then the third part is a nitrogen containing base shown here. And this base can be either a purine or a pyrimidine. And I'll talk about that in just a moment. All right, any question about that? In DNA, the nucleotide base of one strand of DNA can hydrogen bond to a nucleotide base in the corollary strand of DNA. And adenine binds to thymine with 2 hydrogen bonds, one here and one there. All right, DNA. Let's talk about the purines and pyrimidines. DNA has the purines adenine and guanine, and you can remember these as adenine or A, or guanine or G. The pyrimidines are cytosine in DNA and thymine or T. And how an easy way to remember a pyrimidine from a purine is the word pyrimidine has a Y in it, and cytosine and thymine have Y's in the name. Adenine and guanine do not have Y's in them. Any question about that? So this trick works with DNA. With RNA, you have to remember that RNA has the purines adenine and guanine, and the pyrimidines cytosine and uracil, meaning in RNA, uracil replaces the thymine. And uracil is a pyrimidine, but it doesn't have a Y in it. But if you remember that uracil is replacing thymine, thymine has the Y in it. So that's how you can remember your pyrimidines. All right, so the bases bind to each other. And this binding is by hydrogen bonding. A hydrogen bonds with T, unless we're talking about RNA, in which case A hydrogen bonds with U. And we won't usually talk about RNA. And then C hydrogen bonds with G. A and T have 2 hydrogen bonds between them. And C and G, shown here, have 3 hydrogen bonds between them. So that the C and the G is a stronger bond than the A and the T. Any question about any of that? The DNA pairing of one strand of DNA is always paired with the A and the T, meaning the A and the T are complementary bases. And the C and the G are complementary bases. They pair with each other, and they pair because they bind, hydrogen bond with each other. Now, if the only link holding DNA together is a hydrogen bond, and I've already mentioned that hydrogen bonds are very weak, and especially in a liquid like our body, it's mostly liquid, and at our body temperature, which is 37 degrees Celsius, a hydrogen bond breaks and reforms all the time. So if the hydrogen bond in DNA is breaking all the time, why is the DNA held together in a double, in 2 strands held together by hydrogen bonds? Come on, you're seeing it right here. All right, you guys aren't volunteering answers very well. There are many hydrogen bonds in two strands of DNA. And if one breaks, it's not a problem because other hydrogen bonds are holding the DNA together. And then this one will reform and another hydrogen bond will break. And the breaking and the reforming of hydrogen bonds is what we call breathing, because the DNA can open up and then reform. And that happens all the time in our cells because we're at 37 degrees Celsius. Any questions about any of that? All right, what's the sugar in DNA? All right, you guys can look that up. Is DNA double-stranded or single-stranded?
Double?
DNA is double-stranded. And DNA is held together in one strand of DNA by a covalent bond between the sugar and the phosphate. Let me show that. Well, I don't have a good picture of it, so I'll just show it here. And that is, this is the nucleotide, and then the sugar phosphate is a covalent bond holding this single strand of DNA together. So the sugar phosphate is the backbone holding the DNA strand together. And that's different from the two strands, which were only held together by a hydrogen bond, and they're held together by the bases. Anyways, DNA forms a twisted ladder with the bases being the steps of the ladder. And that's shown here. Any question about any of that? I'm trying to finish this. We're going to go on a little bit longer, but hopefully we'll finish in just a few minutes. RNA, the sugar is ribose. And does anybody know, is RNA single-stranded or double-stranded?
Single?
Single.
Yes, in our cells, RNA is single-stranded. RNA, the single strand of RNA is held together by the sugar phosphate backbone. The bases are adenine or A, uracil or U, cytosine or C, guanine or G. There are three major kinds of RNA: transfer RNA, which is tRNA, messenger RNA or messenger or mRNA, ribosomal RNA or rRNA. And if the nucleotides are the same in tRNA, messenger RNA, or rRNA, what is the difference between tRNA, messenger RNA, and rRNA?
They do different things.
Well, that's true. They do different things, but I'm trying to say, what's the difference in their makeup? Do they have the same nucleotides?
No.
Guess again.
Yes? Oh, they
They
have
have
the,
the same nucleotides.
yeah, they
The
do.
only difference in tRNA, messenger RNA, and rRNA is the sequence of those nucleotides. Okay, so tRNA, messenger RNA, and rRNA is the same molecule. It's just to be tRNA, you have to have a specific sequence of the nucleotides to be tRNA. And to be rRNA, you have to have a specific sequence to be rRNA. And to be messenger RNA, you have to have a variety of different sequences to be messenger RNA. So they're the same molecule, they just are different sequencing of the nucleotides.
mRNA is what's in the COVID vaccine, right?
For two of the vaccines made by Pfizer and Moderna, it is messenger RNA, yes. But for Johnson & Johnson, it is not.
Hmm.
Okay. Johnson & Johnson is a protein vaccine. All right. ATP is adenosine triphosphate. It is a nucleic acid, and actually it's a nucleotide, the A nucleotide. It is used as the primary energy source for cellular activities. It simply has adenine, ribose, the sugar group, and then 3 phosphates attached to it. Note that this phosphate link right here and that phosphate link right there are unstable and high energy bonds. When the cell can break this bond when it wants energy, and if it needs more energy, it can break that bond. And these 2 bonds release a lot of energy. You should know that ATP is the energy, what's the wording I'm using? The energy source. Whenever cells want energy, they will get it by breaking ATP. So you break off this bond, you simply add water, and the reaction goes this way. You get ADP, adenosine diphosphate, an inorganic phosphate, meaning a free phosphate, and then energy is released. And if the cell needs the energy, it breaks ATP generally to get the energy, and then use that energy for the cell's needs. The cell makes ATP by dehydration synthesis, so it gets ADP and an inorganic phosphate, and you have to supply energy into this system. And then the reaction goes this way, forming water and ATP. And so this reaction is reversible. It can go either way, and the cell goes this way when it needs energy, and then it wants to make ATP, it goes this way. All right, here's a question for you. We've talked about hydrogen bonds, and they give molecule important properties, like in the bonding and the forming of the protein. What would happen to these molecules if all of the hydrogen bonds were to disappear? So in the world, tomorrow, all the hydrogen bonds disappear. What would happen?
Dry out.
I suppose if we're talking about water, the hydrogen bonds link the water molecules together to get liquid water. And so if you lose all the hydrogen bonds, you'll now have gaseous water. And so there will be no more liquid water on the planet. There would be no more functional protein. So all of the proteins would be denatured. Carbohydrates have hydrogen bonds in them as well. So those would break. So life as we know it would not work. And obviously the earth would no longer be an environment where life could live. We no longer had liquid water. Just telling you how important hydrogen bonds are. All right, any questions about any of chapter 2? Okay, no questions. I'll see you at 6:30 for the lab.